So thank you for the invitation to speak today, and I'm delighted to tell you about our work focusing on the genetic changes in human pluripotent stem cells. Why do we focus on the genetic changes? Well, as we all know, human pluripotent stem cells represent an excellent source of differentiated cells, be it for studies of basic biology, disease modeling, or indeed applications in regenerative medicine. However, most of these applications require the production of a large number of differentiated cells. If we take cell therapy of the heart as an example, there it is estimated that we may need as many as 10 to the 9 cardiomyocytes just for a single transplant. And what this really means is that to generate large numbers of differentiated cells, we may need to expand large numbers of undifferentiated stem cells and keep them in culture for long periods of time. And all the meanwhile, our stem cells are susceptible to the acquisition of genetic changes, aberrations, and these aberrations can then have profound functional consequences, first of all, on the ability of stem cells to generate desired differentiated cell types, but also on the maturity and functionality of those differentiated cells. And then, most concerningly, there is a worry that these genetic changes that are acquired in the stem cell state could confer tumorigenic or malignant properties upon transplantation into patients. Now, what we know about genetic changes that are acquired in pluripotent stem cells upon culture is, first of all, that they appear at all levels. So we see everything from whole chromosome gains and losses down to single nucleotide variants. However, when we look across multiple samples, multiple lines, cultured in different labs across the world, then we see that this pattern of aberrations is not entirely random. At the karyotype level, we tend to see the same karyotypic aberrations time and time again, and these are gains of chromosome 1, in particular 1q, chromosome 8, 12, 17, 20, and X. Similarly, at the single nucleotide variant levels, we recurrently see mutations in cancer-related genes such as TP53. And this is again a worrying parallel between aberrations that on the one hand are seen in pluripotent stem cells upon culture, but on the other hand are also prevalent in cancer cells. Now, what is this recurrency, this non-random pattern of aberrations in hPSCs telling us? Well, it's telling us that we have this kind of a model of essentially evolution playing out in vitro, whereby stem cells acquire mutations, aberrations, but then it's those variant cells that possess selective growth advantage that go on to outcompete wild-type cells and dominate cultures with their own progeny. So these are the recurrent aberrations because they provide selective growth advantage to our stem cells. And so what this means is that if we want to minimize the appearance of variant cells in culture, if we want to de-risk the process for basic research and for cell therapy, we need to understand both how mutations arise in variant cells in the first place, and then how are they eventually selected for, or how they impact cellular fitness and fate decisions in pluripotent stem cells and their progeny. And essentially, these are the questions that my lab is tackling, and I will tell you a little bit about our work in each of these topics. If we start with the process of mutation, so one aspect that we are exploring here is to what extent are pluripotent stem cells really susceptible to things like genome damage or mitotic errors? And also, to what extent does this appearance, for example, of double-strand break predispose them to specific karyotypic aberrations that we see? So if we look at the recurrent genetic changes from this heat plot that I already alluded to, we see that certain aberrations involve gains of whole chromosomes. For example, trisomy 12 is most commonly acquired as a gain of a whole chromosome. And this is presumably due to errors during mitosis or chromosome missegregation errors. But many of the recurrent aberrations that we see in the genome of pluripotent stem cells are actually structural variants, things like translocations, for example. And these are most likely underpinned by double-strand breaks followed by erroneous repair. So the first question we had about this is, to what extent are pluripotent stem cells susceptible to the acquisition of double-strand breaks? And to answer this question, we use the model whereby we take differentiated cells, reprogram them to an induced pluripotent stem cell state, and then differentiate again. And this is a really nice model because here we have isogenic backgrounds, the same cells that just exist in different states, either pluripotent or differentiated. And when we looked at the appearance of double-strand breaks across these cell populations, either by looking at gamma H2AX as a marker of double-strand breaks or doing more specific COMET assays to look at genome breakage. In either case, we see that specifically it is the pluripotent state that is particularly susceptible to double-strand breaks. And following on from this, we asked, well, is there a particular pattern to where these double-strand breaks appear in the genome of pluripotent cells? And to answer that question, we took 4 different pluripotent stem cell lines. 3 were embryonic stem cell lines and 1 induced pluripotent stem cell line. And then we also pushed them out of pluripotency. So similar to this, we have pluripotent stem cells and their differentiated counterparts. Then we subjected them to a method called induce-seq, which allows mapping of double-strand breaks at the nucleotide resolution in the genome. Now, doing this across 4 lines and the differentiated counterparts told us, first of all, as we saw previously, that there's way less genome damage in the differentiated state compared to the pluripotent one. And secondly, when we clustered these samples, performed the clustering, we could see that they separate based on whether they are pluripotent or whether they're differentiated. And this was important to us because it's telling us that it is cell state rather than the genomic background that determines the types or the amount and the location of genomic double-strand breaks in the genomes of these cells. So following on from this, we decided to look more specifically at where exactly in the genome these hotspots occur. And for this, we first of all calculated the break coverage in all of our samples, then thresholded them to identify regions that have that are particularly enriched for double-strand breaks, subsetted them such that we find those double-strand breaks that occur in at least 2 or more samples, and from that determined that we have about 634 breaks or hotspots that are preferentially enriched in the pluripotent versus differentiated state. Now, looking a bit more in detail into these hotspots, we first asked whether there are any specific genomic and epigenomic contexts that could tell us why they appear in these particular sites in the genome. And for this, we combined our induce-seq data with published studies that showed sequencing for example, DRIP-seq for R loops or ChIP-seq for different histone marks, RNA-seq for transcriptomics, etc. And overlaying all of that data, we do see that hotspots in the pluripotent stem cell genome tend to occur in sites that are transcriptionally active and associated with open chromatin. Specifically regarding transcription, we do see that pluripotent enriched hotspots overlap pretty much entirely, overlap expressed genes, and that those expressed genes are in fact expressed at higher levels than genes that are overlapping differentiation enriched hotspots. And finally, these pluripotent enriched hotspots also overlap early replicating sites. This is quite curious because in many other cell types, hotspots of genome damage associate with late replicating sites, such as many common fragile sites, in fact, are in these late replicating genome regions. But in some cancer cells, there are also hotspots that associate with early replicating sites of the genome. So from this, we concluded that there are certain features in the pluripotent stem cell genome that predispose those sites to frequent breakage and double strand breaks. But do they actually correspond? Do they correlate with sites where we see recurrent aberrations in these cells? So to look at that, we first of all took a heat map of recurrent genetic variants, and from that heat map we could, and the karyotyping data that we have, we could infer where the recurrent breakpoints of these structural variants occur in the genome. So in this case, highlighted in red here is a breakpoint from the karyotyping data that is seen on chromosome 1q. And then we took these recurrent breakpoints and basically looked whether they associate with our breakpoints, hotspots that we see in the induced seq data. And overall, we do see enrichment. So we can see that hotspots do to some extent underpin recurrent breakpoints that are eventually seen in the karyotyping data. But of course, this is not a complete picture. Not every recurrent hotspot in the induced seq data really results ultimately in karyotypic aberrations. So overall, what this work has shown us is that, first of all, pluripotent state confers increased levels of genome damage, and we think this could be due to their specific cell cycle architecture, because these cells are cycling faster than their differentiated counterparts. There is correlation with recurrent genetic changes and double-strand break hotspots in the genome. However, genomic double-strand break hotspots do not entirely explain the prevalence of all recurrent abnormalities that are eventually seen in karyotyping data sets. And the reason for this is, of course, that the other side of this coin is selection. So only those cells, only those variants that possess selective growth advantage will go on and prevail in culture. And this is why in my lab, we also have a very firm focus on selection. And today I'm going to tell you a couple of examples of how we go from understanding or noting what are the recurrent karyotypic aberrations in culture to drilling down into the molecular and cellular mechanisms that drive their selective advantage that will ultimately provide us with a platform for stopping their occurrence in culture. So the first of this example is really referring to changes in trends of recurrent genetic aberrations over time. So we know that, for example, since human embryonic stem cells were first derived back in 1998, they were first derived on feeders in the presence of serum or knockout serum replacement. But then in the last 20-something years since they were first derived, the whole field has pretty much shifted to the use of feeder-free, serum replacement-free media. And the question we had was whether this shift in culture conditions has influenced the recurrent aberrations that we commonly see in human pluripotent stem cells. And so to answer this question, we took first of all, we took really extensive karyotyping data sets that we obtained in collaboration with WiseCell, a major stem cell bank in the United States. And we performed this type of analysis where we took 6 most common karyotypic aberrations in the data set and plotted these aberrations over time, so with respect to the year in which they were detected. And what this showed us was a really striking kind of graph that exemplifies that these aberrations are not constant. In other words, that we do have changes in trends of genetic aberrations over time. If we look back in 2009, over 60% of all karyotypic aberrations in the Y cell dataset were gains of chromosome 12, trisomy 12. Very few of them were gains of chromosome 20, and really none of them were gains of chromosome 1q. But if we look in more recent years, then the situation is entirely changed, because nowadays, trisomy 12 is not as prevalent anymore, and instead we have many more aberrations involving gains of chromosome 20q, either 20q CMV or isochromosome 20q, and also chromosome 1q gain, which really was not very common in the early years of hPSC culturing, is now one of the most common aberrations. Similar situation is seen in our in-house data set in Sheffield. So here, back in 2002 to 2004, about 50% of our aberrations, or 50% of karyotypes that were abnormal, contained gains of chromosome 17, and the other 50% were made up of gains of chromosome 12. But in recent years, we pretty much don't really see gains of chromosome 17 anymore. And instead, the most insidious aberration in karyotypes in Sheffield is this gain of chromosome 1q. Now, that was really curious, because we knew exactly that in the lab from our sort of metadata, we knew that this switch in the trends of aberrations kind of coincided with the time when the lab moved from using feeder-based to using feeder-free conditions. And really the hypothesis that came out from looking at karyotypes in this way is that perhaps back in the early 2000s, gains of chromosome 1q were not prevalent, because 1q gain does not provide stem cells with selective advantage on feeders. But they are prevalent nowadays, and in fact, so common because gains of chromosome 1q provide strong selective advantage to cells cultured in feeder-free conditions. So this was, in fact, a hypothesis that we went on to test experimentally. And to do that, we took 3 different lines, 2 embryonic and 1 induced pluripotent stem cell line. And for each of the diploid lines, we also had same cells, but with a gain of chromosome 1q. So we have clonal lines, and we performed the so-called mixing assays, whereby we take these 1q cells that are fluorescently labeled, and we mix them in with their wild-type counterparts, and then perform subsequent passaging and look whether they prevail in culture, which would mean that they have selective advantage, or do not prevail in culture, retain the ratio, which would mean they do not have selective advantage. And the conditions that we tested for this were growing these mixed cultures either on feeders or feeder-free. Strikingly, and entirely in line with our hypothesis, we found that 1q cells, in fact, won and dominated the cultures, but only when they were grown feeder-free. And the same cells grown on feeders showed no selective advantage. This was a very robust result, which held true across all 3 different pairs of lines that we tested. So consistently, we saw that 1q cells win when they are grown on feeders, but the same cells do not have selective advantage when they are moved onto feeders. So 2 questions we had following this result. First of all, how exactly do 1q cells win over wild-type cells when they're grown feeder-free? And 2, why is it that these seemingly very strong selective advantage that 1q cells have in feeder-free conditions no longer applies when the same cells are moved onto feeders? And so to answer the first of these questions, i.e. what underpins selective advantage of 1q cells in feeder-free conditions, we performed a series of phenotypic assays, including these so-called time-lapse clonal analysis assays, where we plate wild-type cells and 1q counterparts in feeder-free conditions. And we're essentially looking at the ability of these single cells to reinitiate colonies. We perform lineage tracing so that we can see when each cell divides, as indicated by this kind of horizontal line, when it gives, so from that we can also infer the cell cycle time, etc. And the red crosses in these lineages indicate cell death. So we can see just from the number of red crosses in wild-type cultures versus 1q cultures that in fact there's way more apoptosis going on in our wild-type cells, and they're not as robust, they do not generate stem cell colonies as readily as the 1q variant counterparts. So 1q cells did have a slightly faster cell cycle time, which also gives them a bit of an advantage in culture. However, the major advantage really comes from the reduced apoptosis. In other words, all three lines showed less cell death, as I said, evident from the having low number of these red crosses in lineages, but also when we quantify this looking at the marker of apoptosis, cleaved caspase, in all three lines, we consistently see that variant 1q cells show less apoptosis than their wild-type counterparts. At the molecular level, we were interested in what is the driver gene for this striking phenotype. And for this, we summarized all of the karyotyping data. So we had, as I said, many thousands of karyotypes, many of them with a gain of chromosome 1q. So by looking at all of those karyotypes, we were able to identify a region on chromosome 1q that is common, so minimal amplicon, minimal region that is always common in 1q variants. And that region contained a number of genes, but one that really stood out to us is MDM4. And it stood out to us because MDM4 is a negative regulator of p53. We know that p53 is really important for stem cell biology, and we also know that mutations in p53 are recurrent in pluripotent stem cells. So it really provided or proved to be a good candidate. Looking at the expression of MDM4 by Western blotting, all the 1q lines had higher expression of MDM4 compared to their wild-type counterparts, and this is as expected because all these lines have an extra copy of chromosome 1q and therefore an extra copy of MDM4. We performed functional analysis doing knockdown in variant cells and saw that this indeed depletes their advantage, and conversely MDM4 overexpression wild-type cells exactly phenocopied variant 1q cells. So we're quite satisfied by this, that really MDM4 on chromosome 1q is the driver gene that provides pluripotent stem cells with selective growth advantage in feeder-free conditions. But why is the MDM4 mediated gain not effective when cells are moved onto feeders? And a really telling experiment here was looking at the expression, or rather localization, of MDM4. This is because when we stained for MDM4 by immunocytochemistry, we found that MDM4 in feeder-free condition localizes in the nucleus in this kind of punctate manner, whereas MDM4 on feeders has a very diffuse localization across nucleus and the cytoplasm. And this was telling because we knew from published studies that, in fact, this type of MDM4 localization is associated with its role in genome damage. So indicating that perhaps pluripotent stem cells in feeder-free conditions experience higher levels of genome damage. And we went on to test this and quantified the levels of double-strand breaks in feeder-free versus feeders. And in line with what we saw with MDM4, indeed, we can tell from this quantification that pluripotent stem cells grown in feeder-free conditions exhibit higher levels of genome damage compared to the same cells grown on feeders. And importantly, when we deliberately induced genome damage by treating cells with compounds, chemicals that induce double-strand breaks in the genome, then we see that wild-type cells readily undergo apoptosis, but in fact, variants with the gain of chromosome 1q or wild-type cells that overexpress MDM4 are now resistant to that genome damage-induced apoptosis. So overall, in this work, in this study, we went from identifying the difference in 1q gains, prevalence of this 1q gain in feeder-free versus feeder conditions. That told us that the reason for this discrepancy is because there's higher genome damage in feeder-free conditions, and 1q cells overcome that specific selective pressure, and therefore this is why 1q cells or 1q gains are more prevalent in feeder-free compared to feeders. This we think is now a platform to improve culture conditions and provide better conditions for growth of wild-type cells so that 1qs do not have as strong of a selective advantage anymore. Another example is really or revelation that sometimes it is not enough for cells to have just faster cell cycle time and reduced apoptosis, but variants can also be very aggressive and use a kind of non-autonomous cell-cell interactions to gain their clonal dominance. And really this was revealed in a study we've done a few years ago where we mixed wild-type cells labeled here in red with their variant counterparts labeled in green. We mix them and observe how these cells behave over several days in culture. And what we see is that, in fact, in this type of interaction, variant cells corral wild-type cells and then effectively eliminate them from culture. And we went on to characterize this process. It's akin to phenomenon of cell competition that's been described in many other developmental and cancer biology contexts. And we systematically assess the type of cell competition that could be going on in our culture. So we, first of all, through transwell experiments, show that it is not trophic interaction, but in fact, we have a so-called mechanical cell competition, which we proved by looking, for example, at how these cells interact when they come in contact at a specific border. So from these kinds of time-lapse experiments, either wild-type cells with their wild-type counterparts or wild-type and variant cells, we see that variants are very aggressive and they push into the space of wild-type cells and really displace them from the joint border. Importantly, where we find the apoptotic signal is not just at the border, but many cell diameters away, telling us that the type of competition that we see of this negative interaction does not only impact wild-type cells in contact with variants, but also wild-type cells many cell diameters away because it happens through mechanical contact. And we went on to characterize the molecular mechanism for this and show that, in fact, these two cell population, wild-type and variant, despite being sublines of the same line with and without genetic changes, behave very differently or respond very differently to each other and to the same mechanical environment in which they are found, with wild-type cells sequestering YAP into the cytoplasm and subsequently undergoing apoptosis, whereas the surrounding variant cells that they're in contact with still retain YAP in the nucleus and can survive and dominate the cultures. This was a really important revelation for us because it told us that in thinking about how variants dominate cultures, we need to think not only about their cell autonomous properties, cell cycle time, apoptotic rate, but also how they interact with their wild-type counterparts and how they can exert their dominance by influencing how wild-type cells behave in culture as well. And so with that, just to summarize, in pluripotent stem cells, we see recurrent genetic changes which can impact behaviors of cells and can be detrimental for cell therapy. We know that now that culture conditions drive the appearance of specific variants, and that variant cells can, in addition to cell autonomous properties, use non-cell autonomous signals to achieve their clonal dominance. And with that, I would just like to thank everyone who's done this work, members of my lab, also our funders, and thank you very much for your attention.